This lecture covers the structure and function of proteins. Proteins have many different functions. Some of them are part of the structure of the body, like your hair, nails, and muscles. Some of them are hormones, like estrogen and testosterone. Some are antibodies working to defend your body. Some play an important role in transport, like hemoglobin in the blood, which transports oxygen around your body. And some are enzymes, which are molecules that speed up reactions. These are a couple of the characteristics of proteins. First of all, they're found in foods like meats, peanut butter, cheese, and beans. And they are composed of amino acids. These are the building blocks, or the monomers, that are made up of carbon, hydrogen, oxygen, and nitrogen.
This is the basic structure of every amino acid. There are 4 bonds around the carbon atom. One bond goes to an NH2 group or an amino group, one goes to a hydrogen, one goes to a carboxyl or acid group, COOH, and the last bond is different in each of the 20 amino acids.
There are 20 different amino acids, and we divide them into 2 groups, depending on if the body can make them or not. 11 amino acids are non-essential, which means the body can make them, so we don't need them in our diets. 9 amino acids are essential, which means that we cannot make them, and we must be sure to include them in our diets. When amino acids bond and form a long chain, a protein is produced. The bond between 2 amino acids is known as a peptide bond, and you can see that in the structure to the right.
Enzymes are proteins that speed up reactions. They act as biological catalysts and have a globular or round shape.
Enzymes are proteins that act as biological catalysts and speed up a reaction. When we talk about enzymes, there are 2 very important terms that we should know. One is substrate, which is the starting molecule. It's the substance upon which the enzyme works. And the second is active site, which is the place the substrate sits on the enzyme so the reaction can take place. You can see the relationship between the substrate, active site, and the enzyme in the diagram on the right. Remember that enzymes are molecules that speed up a reaction. They act as a catalyst. Most, although not all, enzymes are proteins. Enzymes work by binding to substrates and holding them in such a way that the chemical bond breaking and bond forming processes can take place more readily. Once this has happened, the enzyme releases what's now the product and returns to its original shape. So this enzyme can catalyze other reactions just like this one. Here are some characteristics of enzymes. I mentioned already that at the end of a reaction, an enzyme will resume its original shape as it had when it first entered that reaction. Enzymes are usually globular or spherical in shape, and they can be destroyed by high temperatures, strong acids, and strong bases. An enzyme lowers the energy required for the reaction to occur. Enzymes are specific, which means that an enzyme can catalyze only one type of reaction and not many different types of reaction. Remember, the word catalyze means to speed up a reaction. There are laws that govern energy, and they are known as the laws of thermodynamics. The first law of thermodynamics says that energy can be changed from one state to another, but it cannot be created or destroyed. So that means that the total amount of energy in the universe will remain the same. Take a look at the diagram below, and it may help you to understand it a little bit better. The second law of thermodynamics says that disorder, also known as entropy, in a closed system is continuously increasing. When chemical reactions occur, bonds break and new bonds form, and atoms become rearranged into new molecules, so you have the same atoms, but it's a different arrangement and a different molecule. All chemical reactions require the input of energy to start the reaction. In general, if you make a chemical bond, energy is going to be stored. If you break a chemical bond, energy will be released. If a reaction requires energy, it's said to be endergonic. If a reaction produces energy, it's said to be exergonic. And an example of an exergonic reaction is the breakdown of ATP into ADP plus phosphorus plus energy. Since energy is given off, this is an exergonic reaction. We break down ATP into ADP and phosphorus and energy so that we can do the work around the body that's necessary to keep our body functioning properly. Let's look next at lipids. There are 5 main groups of lipids. They include fatty acids, fats, oils, phospholipids, and sterols, and we will be talking about each of these. To determine if a molecule is a fatty acid, you should always look for 2 characteristics. Fatty acids always have an even number of carbon atoms, and this includes the one in the acid group, and they always have an acid group on one end. Look at the picture on the right. You can see the hydrocarbon chain and the carboxyl or the acid group on the right. Check for both of the characteristics I just mentioned in this particular structure. If you count the carboxyl group carbon, you have a total of 6 carbons. Remember that the carboxyl group is the acid group, so this fits the criteria. There's an even number of carbon atoms, and there is an acid group in this structure. Fatty acids are classified on the basis of 2 characteristics. One is the length of the carbon chain, and the other is how saturated it is with hydrogen atoms. If you look at the molecule below, you'll know this is a fatty acid because it has an even number of carbon atoms, including the carbon in the acid group, and there are as many hydrogens as I can possibly put around these carbons, so it's said to be saturated. There are 3 groups of fatty acids based on the length of the chain. A short chain fatty acid is a chain with 2 to 10 carbon atoms, and an example is butter. If you look to the example on the right, you'll see that it has 4 carbon atoms, so it falls into the short chain fatty acid category. Long chain fatty acids have 12 to 18 carbon atoms in their chain. If you look at the one on the right, you'll see that it has 12 carbon atoms, including the one in the carboxyl group. These are found primarily in animal fats and vegetable oils. The last are the extra long chain fatty acids with 20 or more carbon atoms. They are found primarily in fish oils, and the example on the right has 22 carbon atoms, including the carbon in the carboxyl group. Another way that we classify fatty acids is according to how saturated they are with hydrogen atoms. What you have to keep in mind is that carbon has the ability to form 4 bonds and hydrogen has the ability to form 1 bond. If you look at this molecule and ask yourself, can I put any more hydrogen atoms around it? The answer is no, because all the bonds are filled. So when we have as many hydrogen atoms as possible, the molecule is said to be saturated. There are no double bonds between the carbon atoms. A fatty acid is said to be monounsaturated when it has 1 double bond between carbon atoms, as in the example on this slide. If I were to break that double bond and have a single bond, I could add 2 more hydrogens. So this is not a saturated molecule. Instead, it's a monounsaturated molecule. This slide shows you an example of a polyunsaturated fatty acid. Poly means many or more than one, and in this molecule there are 2 double bonds between carbon atoms. This is just to recap what we've talked about on the last several slides. The difference between saturated and unsaturated fatty acids is the presence or absence of a double bond between carbon atoms. The molecule on the left has no double bonds between carbon atoms. The molecule on the right has 1 double bond between 2 of the carbon atoms. So the molecule on the left is saturated while the molecule on the right is unsaturated, and specifically monounsaturated. So the question you should be asking yourself is, does the degree of saturation matter? And the answer is yes, because polyunsaturated is the most healthy of the 3. The other 2, saturated and monounsaturated, will more readily lead to the production of cholesterol. The next 2 groups of lipids are the fats and oils. The main difference between fats and oils is at room temperature fats are solid and oils are liquid, but they also have some similarities. Both of them have 4 components, 3 fatty acids and a molecule of glycerol at the core. The core of fats and oils is a molecule called glycerol. The structure on this slide shows you what glycerol looks like. The next group of lipids are the phospholipids. Phospholipids are important because they make up cell membranes. Like fats and oils, they have 4 components, but their components are slightly different. They have 2 fatty acids instead of 3, the core of glycerol, but in addition they have a phosphate group. You can see the structure of a phospholipid in the diagram on the right. The last group of lipids are the sterols. They are composed of 4 rings of carbon instead of being long chains of carbon as the other molecules we've looked at so far. Cholesterol is the most important sterol and is the basis of many hormones. Sterols have many different functions. They're necessary for cell membranes. They are used to make vitamin D. They're the backbone of hormones, and they're used to make bile salts, which are an important part of digestion. There are many sources of cholesterol, including eggs, meat, cheese, and even your own liver produces cholesterol. You have two types of cholesterol, HDL and LDL. HDL is your good cholesterol. It's the high-density lipoproteins, and it helps remove other forms of cholesterol from your bloodstream. Higher levels of HDL cholesterol are associated with a lower risk of heart disease. Cholesterol is a waxy substance that's found in all of your cells, and it has several useful functions, including helping to build your body's cells. It's carried through your bloodstream, attached to these proteins. These proteins are called lipoproteins. Your other cholesterol, the LDLs, are the low-density lipoproteins. High levels of low-density lipoproteins can eventually build up in the artery walls and cause your blood vessels to narrow. Sometimes a clot can form and get stuck in the narrowed space, causing a heart attack or a stroke. This is why LDL cholesterol is often referred to as bad cholesterol. These are the desired cholesterol values. A total cholesterol less than 180, an LDL less than 100, an HDL above 45, and triglycerides below 200. If you have a lipid profile done, these are some of the numbers that you would see. I mentioned earlier that cholesterol can be bad, and that's because it's been linked to CHD or coronary heart disease. When deciding whether to include eggs in your diet, consider the recommended daily limits on cholesterol in your food. If you're healthy, consume no more than 300 milligrams of cholesterol a day. If you have diabetes, high cholesterol, or heart disease, limit the cholesterol intake to no more than 200 milligrams a day. Take a look at these three ordinary foods. An egg has 255 milligrams of cholesterol. All of that is in the yolk. This would give you close to your daily intake if you're healthy and more than your daily intake if you have high cholesterol, heart disease, or diabetes. A tablespoon of butter has 12 milligrams of cholesterol. 1 cup of whole milk has 25 milligrams of cholesterol. Another factor that affects blood cholesterol levels is exercise. It's been proven that as you exercise, cholesterol levels decrease. One way exercise helps to lower cholesterol is by helping you to lose or to maintain weight. If you're overweight, it tends to increase the amount of low-density lipoproteins in your blood. And remember, that's the bad cholesterol. This is the kind of lipoprotein that's been linked to heart disease. So the more you exercise, the lower your LDL concentration. Age also affects cholesterol levels. The American Heart Association recommends that all adults have their cholesterol checked every 4 to 6 years, starting at about age 20, which is when cholesterol levels can start to go up. As we age, cholesterol levels tend to rise. Men are generally at a higher risk than women. Stress can affect your cholesterol level. There's compelling evidence that your level of stress can cause an increase in your bad cholesterol indirectly. For example, one study found that stress is positively linked to having less healthy dietary habits, a higher body weight, and a less healthy diet. All of these are known risk factors for high cholesterol. Heredity can affect your blood cholesterol levels, so be sure to choose your parents carefully. We know that about 80% of our cholesterol is made in the body and primarily by the liver. The risk for high cholesterol can increase even more when heredity combines with unhealthy lifestyle choices, such as eating an unhealthy diet. Some people have an inherited genetic condition called familial hypercholesterolemia. This condition causes very high bad cholesterol levels beginning at a young age. We're going to talk very briefly about another group of organic molecules. Remember, organic means the molecule contains carbon. These are the nucleic acids DNA and RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. They're named for the sugars that they contain. If you look at the picture on the right, you can see what DNA looks like. DNA and RNA both contain 4 bases. DNA contains adenine, guanine, cytosine, and thymine, and RNA contains adenine, guanine, cytosine, and uracil. Here is a table listing the most important differences between DNA and RNA. DNA has 2 strands, RNA has 1 strand. DNA contains the sugar deoxyribose, and RNA contains the sugar ribose. DNA contains the base thymine, RNA contains the base uracil. DNA is located in the nucleus and RNA is located in the nucleolus. The job of DNA is to direct protein synthesis, and the job of RNA is to carry out protein synthesis. Be sure you know these differences.